Let's start with Metaphase-I. So during this Metaphase-I, nuclear membrane completely undergoes disintegration. Nuclear membrane is completely disintegrated. Actually, this nuclear membrane disintegration starts by the end of Prophase-I and its disintegration is completed by the early Metaphase-I. So completely it gets disintegrated. Okay. So next event that occurs during Metaphase-I is spindle apparatus formation is completed. Spindle apparatus formation is completed. Actually, the spindle apparatus formation is initiated by the end of Prophase-I and its formation is completed during Metaphase-I. That means that is by the early Metaphase-I. Okay, after formation of the spindle apparatus, the spindle fibers of the spindle apparatus attach with the chromosomes at the point of kinetochores. After attachment, the spindle fibers bring these chromosomes towards the center of the cell, homologous pairs of chromosomes towards the center of the cell. So this is one homologous pair of the cell, this is another homologous pair of the cell. So this is the cell membrane of animal cell. These are the homologous pairs of the chromosomes which are brought towards the center of the cell by spindle fibers. So actually in the spindle apparatus there are two types of spindle fibers. They are continuous spindle fibers and discontinuous spindle fibers. Continuous spindle fibers are the spindle fibers which are running from one pole to another pole continuously. They are called as continuous spindle fibers and these spindle fibers are not attached with chromosomes. Second type of spindle fibers are discontinuous spindle fibers. So discontinuous spindle fibers are attached with chromosomes. Hence they are also called as chromosomal fibers or tractile fibers. Okay. Now these chromosomal fibers or tractile fibers, after attachment with these chromosomes, they bring all these homologous pairs of chromosomes. This is one homologous pair, this is another homologous pair and so on. Like that they bring these homologous pairs of chromosomes or bivalents towards the center of the cell, that is towards the equator of the cell, and align them at the equator of the cell, forming bimetaphasic plate. So what is happening now? Bimetaphasic plate or double metaphasic plate is formed. Bimetaphasic or double metaphasic plate is formed. And the movement of these chromosomes towards the center of the cell or equator of the cell is called as congregation. So these are the changes that occur during Metaphase-I. After Metaphase-I, what happens? Cell next enters into Anaphase-I. During Anaphase-I, homologous pairs of chromosomes undergo disjunction. That means they undergo separation. And these chromosomes, after separation, they start moving towards two opposite poles of the cell. So before starting with Anaphase-I, I want to give some differences between this Metaphase-I of Meiosis-I and Metaphase of Mitosis. So differences between Metaphase-I of Meiosis-I and Metaphase of Mitosis. Right. So during the Metaphase-I of Meiosis-I, bivalents or homologous pairs of chromosomes align at the center of the cell. So bivalents or homologous pairs of chromosomes align at the center of the cell. Chromosomes align at the center of the cell. Align at equator. At the equator. So due to the alignment of this bivalence or homologous pairs of chromosomes at the equator, it results in the formation of bimetaphasic plate or double metaphasic plate. Bi or double metaphasic plate. So now let's see what is the condition in case of Metaphase of Mitosis. So in case of Metaphase of Mitosis, univalence, that means one chromosome, only one chromosome align at the center. Here two chromosomes, but During the metaphase of mitosis, only one chromosome align at the center. So, univalence align at the center of the equator. Align at the center of the cell or equator, which results in the formation of single metaphasic plate, or we can say metaphasic plate. Single metaphasic plate. Metaphasic plate. Okay. Now one more difference is, during the metaphase-I of meiosis-I, each chromosome is attached with only one spindle fiber. So here each chromosome is attached with only one spindle fiber. Whereas in case of metaphase of mitosis, each chromosome is attached with two spindle fibers. Two spindle fibers. Let's see how. Two spindle fibers. Okay. Let's say this is a cell. Inside the cell, this is the plasma membrane of the cell. Inside the cell, these chromosomes are aligned at the center of the cell. Univalents are aligned at the center of the cell, to which two spindle fibers which are coming from opposite poles of the cell are attached. So this is one spindle fiber coming from this pole, and this is another spindle fiber coming from its opposite pole. So like this, each chromosome is attached with two spindle fibers. So these are all the changes that occur during the metaphase-I of meiosis. And we also studied about the differences between metaphase-I of meiosis and metaphase of mitosis. Next, let's start with anaphase-I.